Hello, hi, welcome to our Zootech channel. I hope everybody are safe in this pandemic. Today we're going to do a topic on the genetic material, that is, the DNA is a genetic material. How do you prove that DNA is a genetic material? So, many biologists, despite the earlier experiments of Griff, Avery and others, still believe that protein, not the DNA, was a hereditary material in a cell. As the eukaryotic chromosomes, they consist of roughly equal amounts of protein and DNA, it was said that only a protein had a sufficient chemical diversity and complexity to encode the information required for genetic material. So, however, in the year 1952, this scientist called Hershey and Chase, they finally provided a convincing evidence that DNA is acting as a genetic material. It is the genetic material. So, in the year 1952, American biologist, that is Alfred Hershey and Martha Chase, they set out to determine what composed the genetic material of a bacteriophage. So, their aim was to prove that DNA is a genetic material which is being transferred from parents to the offsprings. Now, we'll talk about T2 bacteriophage. What is T2 bacteriophage? They are the viruses which are infecting the bacteria. They are the virus which are infecting the bacteria. What is the shape if you take? It will consist of a protein. It consists of a protein head, a tail. That is, the protein head will be the genetic material, and then it is contained of a tail. So, this T2 will infect a bacteria called E. coli. This T2 will infect the bacteria called as E. coli, that is Escherichia coli. Now, let me talk the principle behind this. So, DNA will contain phosphorus. DNA will have phosphorus, but it doesn't have sulfur. Okay, DNA will have phosphorus, but it doesn't have sulfur, whereas the proteins, they will be having sulfur but no phosphorus. Okay, so DNA will be having phosphorus but no sulfur, whereas in the case of proteins, they contain sulfur but there is no phosphorus in this. How the process goes, this bacteriophage will attach to your bacterium. When injected to a genetic material, that is the DNA into bacterium, synthesis of DNA replication and the protein coat, tail, they will form a new bacteriophage. That is a process in it. Now we'll see how the DNA is acting as a genetic material. So this Alfred Hershey and this Chase discovered that the DNA is a genetic material of a phage known as T2. That is T2 bacteriophage, we call it as. We'll now see the experiment done by them, how the experiment performed and how they said the DNA is acting as a genetic material, why the protein is not. We'll see it. So, the viral protein and DNA, they were tagged with different radioactive isotopes. Now, taking the protein tagging. Talking about this protein tagging, that is the T2 and Escherichia coli, they were grown in a medium with a radioactive sulfur, that is 35S. Radioactive sulfur 35S, they incorporated only into the phage protein. Whereas this DNA tagging what happened is, they were grown in a medium containing radioactive phosphorus, that is 32P, which was incorporated only into the phage DNA. So what is happening here? The protein labeled and DNA labeled T2 phages, they were allowed to infect separate samples of non-radioactive Escherichia coli cells. So what happened is the cultures, they were agitated to shake the loose phages that they remained outside the bacterial cells. So then what is happening is the mixtures were centrifuged, forcing the heavier bacterial cells into a pellet on the bottom of the tubes. So what is happening? The lighter viruses will remain in the supernatant. So this radioactivity in the pellet and the supernatant, they were measured and they were compared. So what is the result we found is that the Escherichia coli, when infected with a protein labeled T2, most of the radioactivity was in the supernatant with the viruses. In the tubes with the Escherichia coli infected with the DNA labeled T2, most of the radioactivity was in the pellet with the bacterial cells. So when the bacteria containing this DNA phages, that is DNA labeled phages, when returned to the culture medium, the bacteria, they released phage progeny, they contained only 32P in their DNA. Then when bacteriophages containing P, that is the radioactive, that is 35P, they were allowed to infect the non-radioactive bacteria. All the infected cells became radioactive, and in fact, much of the radioactivity was passed on to the next generation of the bacteriophages. So in the conclusion, it clearly indicates that the only DNA and not a protein code entered the bacterial cells. So Hershey and Chase has conclusively proved that it was DNA, not a protein, which carries the hereditary information from virus to bacteria. I will explain you this Hershey's and Chase experiment with the diagram. I will see the bacteriophages, in which I will show you the diagram. The sulfur labeled protein, sulfur labeled protein capsule. Okay, now we'll tell you.
Hold me close till I get up. Time is barely on our side. I don't wanna waste what's left. The storms we chase are leading us, and love is all we'll ever trust. Yeah, no, I don't wanna waste what's left. And on we'll go, through the wastelands, through the highways, till my shadow turns to sun rays. And on we'll go, through the wastelands, through the highways, and on we'll go. Finding life along the way, melodies we haven't played. No, I don't want no rest. Echoing around these walls, fighting to create.
Okay, now we are going to show in the means of diagram how the DNA is acting as a genetic material. So what is this? So this experiment began with the culturing of viruses in two types of medium. So one set of viruses was cultured in a medium of radioactive phosphorus. So whereas another was cultured in a medium of radioactive sulfur. So they observed that the viruses, they consist of radioactive DNA but not radioactive proteins. This is because DNA is a phosphorus based compound while protein is not. The another set of viruses, it consisted of radioactive protein but not radioactive DNA. So the infection for the host for infection was the Escherichia coli, that is the E. coli bacteria. So the viruses were allowed to infect the bacteria by removing the viral coats through a number of blending and centrifugation. So what is observation? What did we observe in this diagram is that the E. coli bacteria which are infected by the radioactive DNA viruses, they were radioactive, but the ones which were infected by the radioactive protein viruses were non-radioactive. So what can we conclude in this is the resultant radioactive and the non-radioactive bacteria inferred that the viruses that had radioactive DNA transfer their DNA to the bacteria, but the viruses that had the radioactive protein, they didn't get transferred to the bacteria. Hence, it is proven that the DNA is a genetic material and not the protein. Thank you.